Looking at the immune system poses the same challenge that the endocrine system did, since the immune system is not located in any one part of the body, but is distributed fairly widely over the body. However, there are multiple places where we can find a definite immune function, and the first one, a little surprisingly, is the skin. So the skin separates us from our external environment, and the entirety of it, known as the integumentary system, has multiple functions, but among those is preventing dehydration, keeping us warm, and making a barrier to the outside world so that pathogens don't have easy access to our bodies. Another surprising place where immune activity takes place is in the bones. The bones are actually part of the immune system. In particular, the long portion of the bone contains a marrow cavity. And the marrow cavity is a place where the hematopoietic tissues are located. Those are the cells that make blood. And in addition to red blood cells, they also make the precursors to most of our white or immune cells as well. Now, the long bones are a major site of hematopoiesis in the young, but that gradually shifts to other bones in the adult, like the sternum and the bodies of the vertebrae. So we can see here that we've got those cavities within, and that is where blood cell formation, both red cell and white blood cell formation, is going to occur to a fairly high degree in the adult. There are many different kinds of white blood cells. We have things called neutrophils, eosinophils, and basophils, but we also have lymphocytes that are going after very specific targets. B lymphocytes release antibodies, and T lymphocytes have many functions, but one thing that's special about T lymphocytes is that they mature in another one of our immune organs called the thymus. The thymus is located superior to the heart and just anterior to the great vessels, and it's kind of a tough to find organ because it looks a lot like the fat that's located in that same area. But the thymus has a right and a left lobe, and it is going to be the place where T cells mature and become cytotoxic T cells, helper T cells, or other specialized types of T cells that fight off specific antigens. Here we can see a histologic preparation of the thymus gland. You can note that it has a connective tissue capsule on its outer surface, and the lobe that we have sectioned here is subdivided into multiple lobules. Now, the main feature that we're going to see in most of these immune organs is that they're going to be the host for a lot of lymphocytes. That's going to be the major cell type we see in a lot of these immune organs. And if we zoom in a little bit closer here, we can see an individual lobule, and within it we have a cortex out here and a medulla lightly staining on the inside. And every one of these dark staining, very kind of dark blue structures inside is actually either a lymphocyte, T lymphocyte, or a thymocyte becoming a T lymphocyte. And the thymus is where they're screened to make sure that they can bind antigens strongly enough to do their job, but don't react with self-antigens and cause any sort of an autoimmune reaction. There are some places in the body that are very exposed to the external environment, and no big surprise, our nasal cavity, oral cavity, where we take in air and food, is a major site where infectious material and pathogens can spread in. So as that material moves into our body, we want to screen for those pathogens, and we have a variety of permanent immune tissues located in this area, such as the pharyngeal tonsil here at the back of the nasopharynx. This is also sometimes called an adenoid, so if you hear that term, it's referring to the pharyngeal tonsil here. On the left and right, we have a palatine tonsil, just here between the soft palate and the epiglottis. And if you ever hear about someone having their tonsils taken out, most of the time they're referring to that removal of these palatine tonsils. There are also tonsillar tissues around the eustachian tube here and the tongue, so we have some lingual tonsils as well. And there can be other smaller areas with tonsillar tissue here, but those are the major ones: the lingual, the palatine, and the pharyngeal tonsils. Now, other immune cells are scattered throughout the gut tube because it's still dealing with the external environment. So all throughout our gut tube we have what's known as MALT. And MALT is mucosa-associated lymphoid tissue. Now, not just the gut, but also the respiratory tract and the urogenital tract, any place in our body that's in contact, even if it's very indirectly, with the external environment, is going to have populations of immune cells that arise and drift away and grow and respond to immune challenges that appear along the way. So we'll find that MALT, again, mucosa-associated lymphoid tissue, in the various parts of the body. One exception to it being not permanent is in the ilium. The ilium, even though we can't see it here, has at its microscopic level a group of immune cells gathered inside it called Peyer's patches or ileal patches. Now these are present throughout the ilium. They're not generally visible from the outside because they're inside the lumen here, but they can often make large bumps on the inside of the lumen that are visible and distinguish it as the ilium. The next immune organ we can take a histologic sample of is one of our tonsils. So tonsils are an example of MALT, mucosa-associated lymphoid tissue, that are permanently fixed in the area that's screening the external environment. This is an example of the palatine tonsil. We can see that we have these germinal centers here, these paler staining circles, and this is where we actually have lymphocytes proliferating. Generally, we're going to find a lot of B lymphocytes in this area, which are going to proliferate to produce antibodies that are going to attack pathogens that the tonsil has sensed moving through the area. Likewise, we have what's called a lingual tonsil here. This is going to be one of the tonsils that is near the tongue, and it's having the exact same process go on. As antigens are encountered by the lymphocytes inside these tonsils, the ones that are activated by noticing an antigen with their surface receptors will then start proliferating and making more lymphocytes, which are going to pump out more antibodies to coat the surface of the antigen and make it easier for the body to degrade and attack. Here we have another example of MALT. This specifically is in the gut tube in the ilium. Now, most MALT present in the gut tube kind of arises, goes away in response to the antigens it encounters. But the ilium, this part of the small intestine, actually has permanent MALT in it called Peyer's patches or ileal patches. And these are going to be, once again, areas where lymphocytes are proliferating. We can see that they actually can proliferate to the point where they stretch outward into the lumen or the lining of the gut there, again creating lymphocytes to attack and moderate the pathogens that are encountered inside the gut tube. So we've seen that there are diffuse immune organs located throughout the body. We've got the skin forming a barrier with the outside. We've got the tonsils and MALT forming a barrier with the linings of the body. But we have other immune organs as well. In addition to the thymus, we've got the spleen here in the upper left quadrant of the abdomen. So here we can see that the spleen is on the upper left side of the abdomen right here, and it has two major functions. One is going to be that the red pulp of the spleen recycles old red blood cells that have gotten brittle or inflexible, and it's going to break them down and send their heme groups and iron to the liver, where they're going to be stored until they need to be used again thereafter. But in addition to the red pulp, the spleen, if we were to cut it open, would have what's called white pulp as well. So the spleen contains white pulp, and that is immune tissue that screens the blood itself for bloodborne pathogens or cells that are infected by viruses, and it's going to remove those cells from circulation, and the B and T lymphocytes that are in the spleen will then mount an immune response to those circulating pathogens. This is a cross-section of the spleen, and like many organs, we can see the spleen has a significant connective tissue capsule on its outside. But one specific thing about the spleen is that it's an immune organ, but also an organ that recycles and screens the blood for old and brittle red blood cells. So it's divided into what's called red pulp and white pulp. Now the red pulp is this very much reddish pinkish area scattered throughout the spleen. But because we've stained this preparation, the white pulp, which actually looks white in a gross section of the spleen, is actually stained dark. So these dark patches here are a bit counterintuitively what we would call the white pulp. And the white pulp consists of lymphocytes that are surrounding nearby blood vessels, and these are going to be screening the blood as it comes into the spleen to make sure that it can mount an immune response to any bloodborne pathogens or any other circulating cells that are defective, whereas over here in the red pulp we have a lot of cells that are going to degrade and break down red blood cells and platelets, releasing heme groups and iron so that the liver can make use of it again in the process of making more red blood cells or for other processes that the body needs. So we've got a way to screen the body for invaders from the outside, invaders in the blood, but the only thing we're missing is a way to screen the intercellular space. And that is the job of a very diffuse group of organs known as lymph nodes. And we're going to be spending some time with the various clusters of lymph nodes throughout the body. But they are going to be screening the lymphatic fluid, which is gathered from the intracellular space. It's going to have a lot of viral and bacterial debris. Antigen-presenting cells can migrate to these lymph nodes, and then again, B and T lymphocytes here can recognize those antigens, mount an immune response, and fight those infections off if they are spreading by way of those lymphatic channels. And here we have a lymph node. Now these lymph nodes are divided into an outer cortex, an inner paracortex, and a much inner medulla. Those are not as distinct here, but what I want you to note is that there are multiple germinal centers here, again, these pale staining areas where we have a lot of proliferation of lymphocytes, and we have lymph fluid coming into the lymph node through afferent ductules. So these little ducts here are bringing the fluid in. It passes into a little space underneath its capsule here, so we can kind of see that little hollowed out space there. It's called the subcapsular sinus. And that lymphatic fluid containing debris from within the body, carried by the lymph fluid, just percolates through the lymph node, exposing itself to the immune cells that are there, giving them a chance to have an immune response before working their way to the medulla, and then out of the lymph node through what is known as an efferent ductule at its hilum here.